Welcome Bio11A to organic molecules, and we start out with an overview of a molecule like methane. So an organic molecule, if it's organic, it contains carbon and hydrogen, okay? Not organic carrots that you get at the store that means that it hasn't been grown with pesticides. It means, in chemistry, if it's organic, it means it contains carbon and hydrogen. So the example of methane here is an organic molecule because it has both. It has carbon and it has hydrogen. So we're going to look at a bunch of different molecules that have carbon and they have hydrogen. They're all the different organic molecules. They look really different, and we'll see examples of all of these. We categorize them, right? All these many carbon and hydrogen molecules into four main types that you've heard because of the food groups that you eat. So those four main types are carbohydrates, proteins, nucleic acids, and lipids. So it depends on how many carbons they have, how many hydrogens they have, what kind of bonds they have, all that, how these are connecting to each other is going to determine whether it's a carbohydrate or a protein or a nucleic acid or a lipid. So what I want you in this course is to be able to recognize one by looking at it. So that's all. I don't expect you to learn how to write them. I don't expect you to memorize numbers or anything that has to do with them, but you can look at it and say, oh, I think that's a carbohydrate. So if I show you a protein, so I think that's a protein. I know it's a protein because it has nitrogen or whatever. I'm going to explain that to you. But recognition. At this level, that's all I need from you is recognition of what these different main types of organic molecules are. Okay, so classic carbohydrates, proteins, fats. You've seen this picture a million times when you talk about food groups. So let's do a little bit of the vocabulary first. A monomer. What's a monomer? What's a polymer? Okay, so a mono is one. Monomer is a single unit of what? Well, it could be a carbohydrate, could be a protein, could be a nucleic acid, could be anything. I'm just talking about one piece, one unit, right? And that unit, usually, in all these different groups, if I put a unit with another unit, and I bead them up into a long chain, I'm going to have a polymer, right? Poly, many, a polymer. So monomer is the one smallest piece, and the polymer is when I chain them all together into one big necklace. So that could be a carbohydrate one, a polymer. It could be a protein polymer. It could be in DNA. It could be a nucleic acid polymer. It depends. But all of them, what they're going to have is a basic unit size bead. Think of it as a necklace. So each one is going to have a bead, and you go adding beads onto that necklace, okay, forming a polymer. So if we start out with carbohydrates, instead of calling it just a monomer of a carbohydrate, we can be more specific and call it a monosaccharide. And then we know it's a monomer of a carbohydrate. Much harder to say. Monosaccharide is easier. So it's the monomer of a carbohydrate. And these are really good examples here. Everything that you've heard of before. Glucose, fructose, ribose, and I can show them to you in different ways. I can just write it out like that. Let's do fructose. I can write it out. Or I can write C6H12O6. That's fructose. But look at this. C6H12O6 is also glucose. So what's different about them? What's different about them is the way that they bond. So this is a hexagon. This is only 5. The carbons are arranged in slightly a different fashion. Okay, so if I show you one single one of these and tell you this is glucose, you know that that's a monosaccharide. You know that's a monomer. Okay. Carbohydrates, though, we're going to take that monomer and we're going to chain it in, like I said, like a necklace and make it long strands of this. And then it's going to be a polysaccharide when we have many of those glucose attached to each other, or many attached also onto a fructose, or long chains of carbohydrates are polysaccharides. And they look really different from each other when you compare them in something like this diagram, which is cellulose, right? Leaf, this is your spinach. And here you have a monomer, 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 and these long chains of monomers. And you eat this. And then your digestive enzymes are going to cut it here and in the middle, cut it between them into monomers. And then your cells are going to use those monomers. But first you've got to eat the spinach or the starch. So in potato, this is under the microscope, it looks like this. And you'd have, these are more branched, do you notice? So they branch in a different way because those bonds are slightly different in starch. And then in your liver, you tend to store sugars also in a form called glycogen, which are long chains of glucose, right? And you're going to cut these into pieces, and you're going to be able to utilize those again in your cells. So these are all examples of polysaccharides, all carbohydrates. So the question is, how do you make these long chains and how do you break the long chains? Because we eat them as long chains, but we cannot utilize them. Our body does not know how to use them unless it breaks it down into the monomer. So you are able to digest a monomer and not a polymer, but we eat polymers and we have to break them down. So that process of make and break and make and break is important in the body. It's important for lots of organisms. So we have a name for that. So when we put two monosaccharides together, we're going to glue them together, we're going to bond them together, we're going to call that dehydration synthesis. Dehydration synthesis. It's a really good word because dehydration means you lose water, and when you do that, you do lose water. And I'll show you how. Synthesis is a good word because it means putting something together. So we're forming something, we're putting something together, and in the process, we're losing water. So look at my picture here. If I didn't want to add here all the different OHs and carbons, and because it's going to be complicated, but I want to show you just one little piece of it. Here it would have an OH on it, and this other fructose molecule also would have a little OH there. And what I want to do is bond these together, but in order to do that, I've got to remove some of those atoms here from the molecule. So from one of them, if I've got a pen here, I'm going to come into blue. From one of them, I'm going to remove the hydrogen from this one, from molecule 1. From molecule 2, I'm going to remove the OH, so an oxygen and a hydrogen. And that is going to allow now for a bond to form there. And it's shown here. It's going to form a bond. So perfect. Now I have a disaccharide because I have two of those molecules stuck together, and I can form something like sucrose, right? In this case, I put glucose together with fructose and I make table sugar. That's sucrose. So I did it how? This was done in the body, could be, or in a plant or wherever. And it's called dehydration synthesis, putting it together. Then I eat this sucrose and my cells say, well, I can't deal with that. I need you to give me either the fructose or the glucose, but I can't have this sucrose thing. So now you're going to have to cleave this. You're going to have to break it again. We formed it, now we're going to have to break it into its two component portions. The glucose and the fructose. That has a different name, breaking. Breaking is hydrolysis. Lysis is to break. And hydrolysis is breaking water. And why are we breaking water? Because actually we took water away when we formed this disaccharide. And if we want to go back the other direction, we have to give the water back. But in order to give it back, we're going to have to break it. We're going to have to break it and put OH in one part and H in the other, and that would be broken, right? I'm going to have a piece of that water in one and a piece in the other. So that's hydrolysis. That separates a disaccharide into a monosaccharide. Okay, so join dehydration synthesis, break hydrolysis. Join dehydration synthesis, break hydrolysis. And the next section goes on to proteins, and that is another video that you have. So we want to end right there. Take care. Thanks for listening.